Assalamu alaikum everyone. Let's go over amino acids in this lecture. So you can refer to chapter 3 in Campbell's textbook. So amino acids are the monomers of proteins, just like carbohydrate having monomers like glucose. Now, amino acids are molecules that have common structure, having a central carbon known as the alpha carbon. They also have an amino group and a carboxylic group, and that's why they're known as amino acids. They all have a hydrogen connected to the central carbon, to the alpha carbon. But they differ in the attachment of R groups, and there are different R groups that can form the 20 different amino acids. Now, the alpha carbon of amino acids is actually a chiral carbon, that is, since it's connected to 4 different groups. Okay, and there's one exception, and that is an amino acid known as lysine. Now, since 19 of these amino acids have a chiral carbon, you can have stereoisomers, that is the D and the L amino acids. Now, an important note is that amino acids that occur in nature are the L amino acids. Now, the difference between the L amino acids and the D amino acid is that, and there are different ways of drawing amino acids, but basically looking at the orientation of the amino group, if it is oriented to the left, then it is an L amino acid. If it is oriented to the right, then it is a D amino acid. Okay. Again, just to emphasize this point, that amino acids occur in nature. The amino acids that occur in nature are the L amino acids. Now, D amino acids can also occur in bacteria, but they do so in bacterial cell walls, maybe some antibiotics, but not in proteins. Okay. So, the R groups of amino acids can be in a chain structure, and carbons can be designated with the Greek letters. So the alpha carbon is the central carbon. This is followed by a beta carbon, gamma, delta, and epsilon. Now, if you have a carbon at the end of the chain, then this is known as the omega carbon. Just like fatty acids, when we talked about the alpha carbon, and then the last carbon is the omega carbon, and that's why you have omega-3 fatty acids, omega-6 fatty acids, and omega-9 fatty acids. Now, so amino acids all have the same structure, but they differ in their side chain or their R group. And they differ in their size, so you can have large amino acids, you can have small amino acids. They differ in their shape. Some are chain in structure, others have ring R groups. They differ in their charge. Some are positively charged, others are negatively charged, and you have many of them having no charges. They differ in their hydrogen bonding capacity, their ability to form hydrogen bonds, since some of them are polar, others are non-polar. And they differ in their hydrophobic characteristic, that is, you have amino acids that are highly hydrophobic, you have non-polar amino acids, and you have polar amino acids as well. And they differ in their chemical reactivity. Some are inert, they do not react at all. Others are highly reactive. So, there are different ways by which we can categorize amino acids. One of them is classification according to their R groups, not the amino acid itself, rather the R group itself. So they can be categorized simply into 4 different groups. You have non-polar amino acids, and these include alanine, valine, leucine, isoleucine, methionine, tryptophan, phenylalanine, proline, and glycine. And you have amino acids that have polar R groups, and these include serine, threonine, glutamine, asparagine, cysteine, tyrosine. We have charged, possibly charged amino acids, lysine, arginine, and histidine. And we have negatively charged amino acids, and these are glutamate and aspartic. So this is what we will do. I'm going to go over these amino acids. You need to know their structures in terms of characterization of these structures of the R groups. Some important notes about these amino acids. You need to know their 3-letter abbreviation. Okay, so let's go over these one by one. Let's start with glycine. Glycine is the simplest amino acid. It's the only one that is not chiral, so we say that it is an achiral amino acid. It's achiral because its R group is basically a hydrogen. So you have a carbon connected to 2 hydrogens, and that's why it is an achiral amino acid. So it's the only one that doesn't have L and D structures. It is a derivative of acetic acid, okay, and it can be considered a derivative of amino ethane. So that's glycine. Now, I noticed that I put glycine sort of like on the side. Now I can put it in the category of amino acids with nonpolar R groups, but I put it on the side because overall, collectively, the whole molecule, the whole amino acid is actually polar, since the hydrogen has minimal effect on the polarity, or the, let's say, the hydrophobicity or the non-polarity of this amino acid. So let's talk about the non-polar aliphatic amino acids, that is amino acids that have chain structures, but they are non-polar. The second simplest amino acid is alanine. The abbreviation is ALA, is AL, alanine is ALA, ALA. So you can name it ALA, you can name it ALA, whatever you want, but you have alanine. Now notice that the R group of alanine is just a simple methyl group. Okay, now maybe I forgot to mention that the abbreviation of glycine is GLY, G-L-Y. So that's alanine. Okay, now then you have 3 amino acids, which are valine, leucine, and isoleucine. Now all of them are branched amino acids, and this branching is important actually. Important when it comes to formation, to protein folding, formation of the 3-dimensional structure of a protein, creating repulsion due to van der Waals interactions. Now the abbreviation of valine is VAL, the abbreviation for leucine is LEU, the abbreviation of isoleucine is ILE. Notice how the branching takes place in leucine and isoleucine, whereas for leucine, the branching occurs at the gamma carbon, the 3rd carbon, that's alpha, that's beta, and this is the gamma carbon. So this is a branch, again, a branch nonpolar amino acid, or you can have the branching for isoleucine occurring at the beta carbon, alpha and beta. Now the 3 of them are essential amino acids, meaning that our body cannot synthesize them, and we have to get them from blood. Now, so the other one is methionine. Methionine can form, methionine is one of 2 amino acids having a sulfur group in their R group. The other one is cysteine. The abbreviation of methionine is MET. We talked about methionine before in the molecular biology course, saying that it's the first amino acid that is inserted in forming proteins during translation. Now methionine forms a molecule known as S-adenosyl methionine, which is a donor of methyl group. So here, this is this section of methionine in the S-adenosine methionine, and this is the methyl group that is donated. Now, another one, the last one, is known as proline. The abbreviation is PRO. Now, notice how the R group actually extends from the alpha carbon, but it goes back and it forms a covalent bond with the alpha nitrogen, forming a secondary nitrogen. This structure, the ring structure, is known as an amino acid. Now, what's important about proline is that it is a very rigid amino acid, so it has very strong effect on protein structure in determining protein structure. Keep that in mind. Alright, so let's go over these amino acids again so that you can memorize them. So we have glycine. Glycine is it's the simplest amino acid, and it is an achiral amino acid. It has a hydrogen as its R group. This is followed by alanine, ALA, having a methyl group as its R group. Then we have 3 branched amino acids. These are valine, val, leucine, leu, and isoleucine, ILE. Notice how the branching of leucine and isoleucine differs, where in leucine the branch occurs at the gamma carbon, whereas for isoleucine the branch occurs at the beta carbon. Then we have methionine, and I said that methionine is one of 2 amino acids having a sulfur group. The other one is cysteine, and methionine can form a structure known as S-adenosine methionine, which is a donor of methyl group. And then we have proline, which is it has an imino structure. It has an amino structure, and it is a rigid amino acid. Its abbreviation is PRO. Okay, so we'll talk about the polar amino acids. And notice that I put glycine here because overall the amino acid itself is a polar amino acid. Alright, so let's go over these amino acids having polar R groups. We have serine and threonine. Both of them have hydroxyl groups in their R groups. Notice how they are related to the structures of alanine and valine. So for serine, instead of a methyl group as in alanine, you have one hydrogen replaced by a hydroxyl group. And for threonine, you have a replacement of a whole methyl group into a hydroxyl group. Now, what's important about serine and threonine is that they are reactive amino acids, and they can be phosphorylated. That is, when they are part of proteins, you can have a phosphate group linked to the hydroxyl groups. Then we have cysteine. Cysteine is the second amino acid that has a sulfur group. Remember, the first one is methionine, but it's non-polar. Now, cysteine has a thiol group, and it's a reactive group actually, since it can form what is known as disulfide bonds, and these disulfide bonds can occur within a protein itself, determining or playing a role in forming the final protein structure, or it can link different peptides or polypeptides to each other, forming quaternary structure of proteins. We'll talk about this later. Maybe I forgot to mention actually that the group in methionine is a thioether group. So it looks like an ether group, but it's instead of an oxygen, you have a sulfur. So I'm sorry. So let's get back. Now the other polar amino acids include asparagine and glutamine. Abbreviations are ASN, and glutamine is GLN, GLN. Both have amide groups as their R groups. Okay, so let's go over them again, these polar amino acids. So we have serine and threonine, or ser and thr, THR, and they are important for protein phosphorylation. We talked about cysteine, and I said that cysteine has a reactive R group. Its abbreviation is cis. It's important for the formation of disulfide bonds. And then we talked about glutamine and asparagine having amine groups in their R groups. So let's go over now another group of amino acids. These are the aromatic amino acids having ring structures. So these include three of them: phenylalanine, tyrosine, and tryptophan. Phenylalanine is rather a large hydrophobic molecule, similar to alanine, having replacing one of the hydrogens in the methyl group of alanine by a benzene ring. That's why it's known as phenylalanine. So it's like a phenyl group attached to an alanine amino acid. Its abbreviation is PHE, Phe. Now you can have a replacement of this hydrogen by a hydroxyl group, creating another amino acid known as tyrosine. The abbreviation is Tyr. Now tyrosine is an important amino acid because first the hydroxyl group makes the amino acid a polar amino acid, and this hydroxyl group is important because it can be phosphorylated. So proteins that are phosphorylated can be so by attachment of a phosphate group to hydroxyl groups of tyrosine and the other two amino acids that we talked about before, serine and threonine. Now the third aromatic amino acid is tryptophan. The abbreviation is TRIP. Now tryptophan is the bulkiest amino acid. It has a double ring structure, again replacing one of the hydrogens in alanine by this indole ring. So this ring structure is known as an indole ring. And notice that this indole ring has a nitrogen, so it makes tryptophan a little hydrophilic, but overall it's a hydrophobic amino acid since the ring structure is large. Okay, so let's go over them again. We have the three aromatic amino acids, and these are phenylalanine with a benzene ring attached to one of the that replaces one of the hydrogens in the methyl group. And then we have tyrosine, where we have a hydroxyl group linked to the benzene ring, and it can be phosphorylated. And we have tryptophan, a bulky amino acid having a double ring structure. Okay, so let's talk about the charged amino acids, and there are two categories. We have positively charged amino acids, and we have negatively charged amino acids. Now, in terms of the positively charged amino acids, there are three of them. Two are lysine and arginine. Now, lysine, the abbreviation of lysine is lys, and it's a chain structure that ends with an amino group. And we have the arginine, abbreviated as arg, having a guanidino group. Now the third positively charged amino acid is known as histidine. It's abbreviated as his. And histidine has an imidazole ring. And notice that this nitrogen right here can be charged. The importance of histidine is that the pKa of this group, of the nitrogen group here, is close to physiological pH. That is, it's around 6. Okay, so this has important physiological implications. Now we have two negatively charged amino acids, and these are aspartic acid and glutamic acid, or aspartate and glutamate. Aspartate is abbreviated as asp, glutamate is abbreviated as glu. Both of them have carboxylic groups at the ends of their R groups. So we have 5 charged amino acids. 3 of them are positively charged, including lysine, arginine, and histidine. And we have 2 negatively charged amino acids, and these are aspartic acid and glutamic acid. So questions in the exam can be something similar to 2 amino acids are negatively charged. The following amino acid is achiral. So we will go over the characteristics of these amino acids in terms of their structure, reactivity, hydrophobicity, and so on. So these amino acids can be derivatives of important molecules in our body. So not only do they make up proteins, but they are important for the synthesis of porphyrins, such as the He molecule in hemoglobin, neurotransmitters, purines and pyrimidines of DNA structure. They can also be used in metabolism for the synthesis of ketone bodies, fatty acids, steroids, or the formation of glucose in a process known as gluconeogenesis. We'll cover that in the metabolism course, inshallah. Now, so we will go over some of these amino acids and their derivatives. So, starting with tyrosine. Tyrosine is a derivative of 3 important neurotransmitters, and these are dopamine, norepinephrine, and epinephrine. Epinephrine is the same as adrenaline, that is responsible for the fight or flight response. Now, these 3 neurotransmitters are known as catecholamine neurotransmitters or catecholamines, since they have this catechol moiety as part of their structures. You are not responsible for memorizing this pathway or for memorizing the enzymes responsible for the synthesis of each one of them. Okay, just the structures of these neurotransmitters. You're only responsible for knowing that they are derivatives of tyrosine. Okay, so tyrosine is also a precursor for 2 other important molecules. One of them is melanin. Melanin is responsible for skin color. And there are 2 types of melanin. You have the eumelanin that gives the dark skin color, and you have the phenylalanine that gives the red color, red hair and red skin. Okay, now of course Africans have a high amount of melanin in their skin relative to Northern Europeans. Now, tyrosine is also important for the synthesis of the hormone thyroxine. Okay, so which is where you have 2 tyrosine molecules linked to each other, and they are iodinated. Now, sort of like an interesting point here is that usually in breakfast people have cheese. Now, they do so because cheese contains tyramine, which looks like epinephrine and acts like epinephrine. So, and it is made from tyrosine. Now, and that what gives energy to individuals during the morning, and that's why egg is a common dish for breakfast. So another amino acid that is important as a precursor of other important molecules is tryptophan. Tryptophan is this large, bulky amino acid that's abbreviated as TRP. Now, tryptophan is the precursor of serotonin, which is another neurotransmitter that gives a sense of sedation and relaxation and calmness. And that's why people drink milk sometimes in order to sleep, because milk contains a lot of tryptophan, which can then be converted to serotonin and relaxing people, helping them go to sleep. Tryptophan is also a precursor for a neurotransmitter, an important molecule known as melatonin, which controls day and night cycles. And that's why people who travel a long distance, like for example going to the US, for example, they suffer from jet lag, and these people can take melatonin to help them adjust in terms of the circadian cycle or the day and night cycle. Now, histamine is this positively charged amino acid, is the precursor of histamine, and histamine is released from immune cells as a reaction to, for example, allergies as well as inflammation. Histamine is also a neurotransmitter. Not only that it is a a derivative of amino acids. Well, it's important as a neurotransmitter. It's involved in allergic reactions. It contributes to inflammatory response, and it causes constriction of smooth muscles. Glutamate is a negatively charged amino acid. Can be converted into a neurotransmitter known as gamma amino butyrate, or abbreviated as GABA. GABA is an important inhibitory neurotransmitter. It reduces neuronal excitability. Okay, and it must be synthesized in the brain, since the brain doesn't has a blood-brain barrier. And GABA GABA has relaxing anti-anxiety and anti-convulsive effects. Now, glutamate can also be a precursor for a modified amino acid as part of proteins, the protein known as thrombin. So it can be carboxylated, so it's already a carboxylated amino acid or having this R group, but another carboxyl group can be added, and that helps in blood clotting. Now notice that an important point is the vitamin K is important for the process of carboxylating glutamate, and that's why it's important for blood coagulation. Okay, and that's the letter K. That's what it indicates. It indicates coagulation because it's written in German, not in a C, starting with a C. Rather, it starts with a K. Okay. Now, arginine is the precursor of nitric oxide. Nitric oxide is a small molecule. It's gas. It has a huge effect on endothelial cells and blood vessels. It has very short half-life. We'll talk about it later on in the third year, inshallah, in the CNS module. Now, nitric oxide functions in vasodilation. It inhibits platelet aggregation, adhesion. It inhibits leukocyte adhesion. It has anti-proliferative action. It has a wide array of effects on the body. Now, lysine and proline. Lysine is the positively charged amino acid. Proline is this rigid amino acid. Both of them can be modified by hydroxylation. So you can have hydroxylysine and you can have hydroxyproline. And these two amino acids, when hydroxylated, are important components of the collagen structure. We'll talk about collagen later on as well. Okay. Now let's talk about an important derivative of glutamate known as monosodium glutamate. Now, monosodium glutamate is used in the food industry, especially it's added into Asian food. It gives it a nice taste, except that monosodium glutamate causes serious physiological reactions in certain people, resulting in feeling of chills, headaches, dizziness, and it's related to a syndrome known as the Chinese restaurant syndrome. You can Google it. You can read about it. You can read about the side effects that are linked to monosodium glutamate. Some people make a big deal about monosodium glutamate and its effects. So we can ask questions about what's specific about proline, We can ask about, again, questions about amino acids like an acetic amino acid is negatively charged, positively charged at physiological condition. We'll talk about ionization of amino acids later on. We can ask questions about amino acids and their functional groups, their side chains. These are different types of questions that we can ask. Okay, now the average weight of an amino acid is about 110, and that's why we can estimate the size of a protein depending on 110 Dalton, that is, and we can estimate the size of proteins based on the number of amino acids that they are made of. So, next lecture we'll talk about ionization of amino acids. Make sure that you memorize these amino acids, their characteristics, their R groups, reactivity, derivatives, and so on, and prepare yourself for this lecture, ionization of amino